Well, we're really delighted that all of you are here this afternoon. We're going to start by introducing ourselves and then telling you a little bit about how teaching a data-driven modeling course together resulted in a textbook. So we're going to tell you a little bit about that book and how it works with all of us, with both of us. And then in the second session, you'll actually see a lesson from that textbook. So Becky, do you want to say something about yourself?
Yes, I'm Becky Sanft. I've been at UNC Ashville almost 6 years, hard to believe. But I met Anne when I had a position at St. Olaf College for 4 years before coming here. And that's where we met and collaborated on this project.
Great. So I'm Ann Walter. I teach in biology. I've been teaching at St. Olaf for a long time. Like many biology departments pushed by vision and change and other situations, our biology department was really anxious to work with the math department to do something mathy, but we didn't know what that was. Fortunately, Becky wanted to work with a biologist, and our associate dean put us together and allowed us to teach together a course. And that's what we're going to describe to you now. And hopefully I can move the slides. There we go. Whoops, there we are. Whoa, go. Well, why won't it stay? There we are. We're not there. I've had this problem before. Okay, so in a modeling course, it's really important to separate things into parts. And so we spend a lot of time integrating the modeling steps with data collection, and our data collection occurs in the lab or via the literature. Our goal is to address real questions, and we also are very, very cognizant that it's important to reassess what we've learned from doing the modeling, revisiting each step in the modeling. So like all of you, I'm sure know well, describing the system, being very clear about putting it into small bits, the parts, then writing a mathematical description of the system, using data to test and validate, and then of course to sit back and look at this and decide whether or not the model has anything to do with the reality that you're looking at is really important. So then, when we were teaching this class, and I am so sorry, our class in our text really fits our situation. We both teach undergraduates, both at UNCA and certainly when we started together at St. Olaf. So we're thinking particularly about undergraduate biology and math students with a minimal math background. And I will note that that minimal math background will work, but most students really need more mathematics. We're also addressing in our textbook any faculty member who's teaching undergraduates in STEM. So it's really an undergraduate beginning modeling course with the real goal of thinking the real world. And that is in typical biological modeling, you're going to have a biology domain expert, a math domain expert, and they're going to work together. The way we have put together exercises, we think that what we have is applicable to beginning courses, especially across disciplines. We've really designed it so students can support each other and learn from each other. We think that our exercises can supplement almost any math or life science course, and you'll see some individual cases and labs with both the biological piece strong and the modeling piece quite strong. And we also, like I think many texts have a good R tutorial, and we think anybody using R in any of your classes might find our tutorial helpful. What you're seeing in this image here is a classic group of students working on projects in the lab. The groups are working on different projects in the lab. We found it's been really helpful to have both a peer teaching assistant and our tutor for our labs. We start the courses, the various courses that we now teach, by thinking about preliminaries. We do not assume that anyone has much experience in R or in lab techniques. And our very first unit will mimic what we do with all of our units. We have a section that involves lots of practice. And I've just named a few of the things that we practice with: matrices, data frames, plotting, etc., all the way through linear regression models that we fit. We don't do this practice in a vacuum, but we practice with real data, and then maybe answer a few small questions. We look at life expectancy versus birth year, ask whether it's linear. We learn to transform data and looking at allometric scaling and a linearized power law using logs of the data, for example. And then because we do have a wet lab in our course, we don't assume any biology, we don't assume any lab skills. So our very first lab is learning how to do stuff and to actually measure things in the lab. And once we've done these kinds of things, we are ready to go on to actually doing some modeling.
We cover 3 units, unit 2, 3, and 4. And starting in unit 2, right away, we have students working through the modeling process of formulating the model, fitting parameters, analyzing the model. We try to keep the math really simple in the beginning because they're learning R, some of them aren't comfortable in the lab. So we first use discrete time equations because those seem pretty intuitive to the students. And with parameter estimation, we're just doing linear regression. We also emphasize qualitative diagrams like you see here, and things like just sketching relationships, kind of how do you even start to think about modeling? And these qualitative diagrams really seem to be the common language between the mathematicians and the biologists. So we really emphasize that throughout the course. So each unit has a background section that goes through a lot of practice, and then we have a culminating exercise at the end. So this one is on whooping cranes, where they go from the start, from formulating the model through making a prediction in this case. And that's the other thing. We also emphasize the uses of models, for understanding, predicting, and control. So once they get a lot of background and practice with these tools, then we jump into case studies and a wet lab. And so you would likely not do all of these. You might pick 1 or 2 case studies and then the lab if you're doing the lab. And so we hit different areas of biology here. We have a case study on island biogeography, pharmacokinetics. So we look at consuming caffeine and looking at caffeine blood levels. And we look at invasive species. Here it's garlic mustard. That's a picture from behind my backyard. And they're using control strategies, trying to figure out, using a model, how can we maintain low levels of this plant? And so the case studies, again, just really go through the entire modeling process. They're using it to either make predictions, thinking about control questions. The lab in this unit is pretty simple. We try to keep these labs cheap and simple for people to run. And here we're looking at E. coli. They're growing E. coli, or you could do yeast. And again, we kind of just start with the data, right? So you look at the data over time. If you plot per capita growth versus population, you see that it looks almost linear. And so that reveals the model that they might try, and it turns out it's logistic growth. And logistic growth is obviously shows up a lot in modeling, and it really is weaved throughout our textbook here. And also one other thing in the labs, that it's typically at least 2 weeks. We meet once a week in the lab at St. Olaf College. And the first week is really guided. We're really guiding them through what they do. But the second week, they get to be a little bit more of a researcher, ask a question and explore that question. The next unit, unit 3, we connect discrete time models with continuous time models here. So we're getting into simple differential equations. We put a lot of emphasis on this unit on the model formulation. So we talk about relative rates, mass action, positive, negative feedback, saturation curve. So how do you take this real world system and translate it to these mathematical relationships? We also investigate parameter space with sliders. The students love sliders. So we can use those sliders to understand how parameters affect model output. We get into nonlinear fits here. And so we talk about how to do that. Model selection, if we have more than one model, how do you compare the two? There are different ways to do this. We use the AIC in this case. And then also here, we have kind of a culminating exercise at the end on temperature dependence of photosynthesis. So they're using a lot of these tools that they've learned. And then the case studies for this unit, we have 3 again in a lab. So we have data on leaf decomposition for different tree species and for low and high nitrogen treatments. And they're also comparing models in this case. So it's a model selection question as well. Tumor growth models, we look at tumor volume data, and they look at 4 different possible models for this, and they're testing both their descriptive power and their predictive power in that case. We look at predator-prey responses, so they're looking at functional response curves of 2 invasive species, and they're also, I should back up. So they're using those to estimate a maximal feeding rate and attack rate. And then we also test some effect on those with temperature, so temperature dependence. In the lab, we look at catechol oxidase, an enzyme that turns apples brown there. And they're using data on this and connecting this to enzyme kinetics. So again, you'll kind of see in the labs, we're touching on ideas that students have likely seen in an intro bio course or other courses, but looking at it from a modeling perspective. So a lot of our students have maybe, at least the ones in biology, have seen some enzyme kinetics. But we're spending a little more time on, seeing where this relation comes up. So here we have the Michaelis-Menten curve, but we start from the differential equations, work through all the assumptions, and understand how do we get to that Michaelis-Menten function. And let's see, the first week, it's really guided. Again, they're just trying to get those constants, that Vmax and Km there. And the second week, they get to try some inhibitors and try to understand what kind of inhibitor they have based on the data they're collecting and the model they're fitting. The next unit, unit 4, we get into more complex systems. And so the last unit, we're looking at really simple differential equations. Here we're getting into systems that we don't have explicit solutions. And so we have to talk about how do you solve these equations numerically. We also do more parameter estimation. It's just a little more complicated. So we use an optimization package in R under the FME package, and they use that to help them fit parameters. We also spend some time on sensitivity analysis. So this is a way to identify which parameters have the greatest effect on the model output. And we talk about how this can be used for research design, right? More effort should be devoted to ones that are really have a great effect on the output. We also talk about it as a tool used in studying control strategies. And so we'll see an example of that in the next session with the case study we go through. And then this one, we also have a culminating, in the background, we also have a culminating example at the end with Ebola virus. And so again, they're going kind of from the beginning, all the way through the modeling steps, from framing the model all the way through sensitivity analysis. And then in the case studies, we look at the 2009 H1N1. We have some data from Australia, and so they use the SAR model to fit parameter values. And then they're assigned, right, to think of vaccination strategies. So this has actually been really interesting to go through this year, right, with everything that's happening. We also see a case study on cancer immunotherapy. That's the one that we're going to go through in detail in our next session, if you'd like to see that. And then the last case study is on quorum sensing. And we look at it in the context of bobtail squid here and luminescence. The lab in this unit is so much fun. Students really get into this one. So we have them eat jelly beans or, consume some sort of sugar very quickly. And then they take glucose measurements. And so we kind of go through the model formulation. What's interesting about this one is, they can't measure insulin, but the model gives them a sense of what that looks like over time for insulin levels. All right, now this leads to kind of the culminating project of the semester, because at this point in the semester, we just kind of barely touch on these ideas, but they have enough in their toolkit at that point that they can do some really interesting things. So Anne's going to talk about that.
Right, so either in the lab or in silico with data, students put together a project. It's usually in teams. And we design those teams to be interdisciplinary. I should note that in addition to everything you've heard about up to this point, we do prepare them to pull things together by having some paper writing exercises. So they have written up their labs. They have also done some exercises independently with variations on a theme from a model. So it's not like they have started with nothing. They also get quite the rubric that gets them going. Before they can start their project, we require them to develop a hypothesis, a research plan, a flow chart of what they're going to do. The research plan needs to include what sort of data they'll actually anticipate getting, their data analysis. And once they've collected all the data and put it all together, they end up with an oral presentation and usually a paper on this, missing on our steps. Topics generally are variations from the lab. So you heard that we did a logistic growth lab with bacteria or yeast. So they might pick another bacterial species. They might pick a different organism altogether, that's some kinds of ciliates that you can grow easily. They might do competition experiments. They might vary the temperature. They might vary the food, something, so that they've got to research questions. With the glucose lab that Becky mentioned, they love variations on that one. And there are all kinds of things you can do from varying the kinds of carbohydrate ingested. They love to do exercise and see if blood glucose goes down. Good news is that it rarely does, but it's interesting to watch them try to make that happen. And if you're not doing a lab, or if you're shut down due to COVID, say last spring, what we pick up and do is a research topic, which was typically a variation of the case study topics. Last spring, many of our projects, as you might imagine, had something to do with an SIR model. Some of them even did some COVID modeling, but they also pick up whatever their particular interests are. So they may be, again, competition models among species are fairly popular. So those are the kinds of projects. And we give almost 4 weeks to these projects. So we really expect them to be worked working in teams, bringing their different expertise together to come up with a paper that they can share. So these are some questions we can imagine that you might be asking yourself right now. Oh my gosh, I've never taught a lab before. And we'll just mention, of course, it's not essential. Becky's been teaching this without a lab at UNCA. But it's worth trying something. And even if you're doing a few dry lab things in the classroom, I think students really, really appreciate the messiness of the data. It's an important thing to learn that the data that are collected aren't necessarily perfect textbook data. And they also like collecting their own data. It's easy to find some technical support, and we have lots of alternative labs and dry lab activities at the end of our book, and there are many resources for such things. We encourage you, anyone in a math department, to partner with a biologist. Biologists are anxious to get more math training, so you might find one who will join up. And definitely a bio major as a teaching assistant can make even a single wet lab go very smoothly. We've enjoyed team teaching. Is it necessary? Of course not, but it's fun. The problems come alive with a biologist, and the math is much more nuanced with a mathematician in the room. I've been team teaching this course now with several other mathematicians, and it seems to work. It works really well. We seem to be able to partner pretty easily and find that kind of handshake between expertise and demonstrate that to students. And I think I also just note that the exercises can easily be modified to be done without a team. So of course, it's not necessary. People wonder how we can support team teaching. One of the things is that we've keep the amount of contact hours low, even the lab hours are low, so that the FTE is minimal. Cross-listing, making sure you're meeting general education requirements, make your class important. We suspect you have more questions, but before we do that, we should acknowledge that we would never have really met, at least not met in this context, without our associate dean who played matchmaker to make this class happen. We might not have gotten off the ground without spending a summer with some students who played critical roles in developing the lab component of the course. So Nora Peterson, Megan Campbell, and Lanza Duano. And of course, we had support throughout this project. And there's some references and other things that you should notice at the end here. So we can go to some assessment quotes just in case you're wondering if students like this class, but maybe this is a good time to go on to some questions.
Hey, let's first of all thank you for the wonderful presentation. Let's thank the speaker first, Becky and Anne. Good job. You can do the Zoom clapping for us. Any questions for Becky and Anne?
We can.
You can either type in the chat or you can just unmute yourself and then you can just say a question.
That would be great. And I can stop sharing, I think. Maybe. So I have to escape.
Hi, this is Maliha. I'm a grad student at the University of Waterloo. I just had a question to both of you, I guess. So before the students can get to the case study component and the lab, how much of the mathematical tools are they introduced to in order to be able to formulate a model and like write down what the model can be or describe it? And are they also taught like how to solve systems of equations, and or is that so how much time do they spend practicing that? and as compared to the time spent on the, I guess, more larger problems of actually formulating a problem, an approach?
Mhm. You know, I would say day one, we start formulating models, right? These simple, discrete time models, they can pick up right away. You can start really simple and they can start doing things. The case studies in that unit 2 are really intuitive, at least 2 of the 3. The third one is much more complex, but the island biogeography and pharmacokinetics, right? They understand how to set up these discrete time models right away, I would say. And we, you know, at least the first week is just kind of spent on this R tutorial and getting used to the lab. And then, you know, by week 2, we're jumping into these simple, discrete models. I don't know, Anne, do you want to add anything on that question?
I would echo that exactly. We actually just did the schedule for the spring's course, and the first week will be preliminaries, practicing, and then by the beginning of week 2, we're right away starting to go from little pieces of modeling to actually thinking about a problem. And the students are learning some of the math, some of the coding by doing the model, you know, so they're motivated to figure out how to do a piece that they need to do, because right now it's urgent. So there's a little bit of just-in-time learning. We do some lecturing, and then we also do a fair amount of group work in class as well as homework. There's quite a bit of homework so that students practice.
And I'll just add that, you know, they're going through the modeling process from day 1.
Yes.
It's not like they're learning the tools and then, oh, we can't really apply it until later, right? They're doing this from day 1. Their tools are getting more sophisticated throughout the semester. You can do things really simply at first with just, you know, simple discrete time model and linear regression. And then your question about differential equations, you know, this is very different than a differential equations course. We don't go through techniques to find explicit solutions to them.
Okay, that's kind of what I was wanting to ask. Like, do they get any practice with like pen and paper solutions, or is it just?
They don't. You know, in the unit where we introduce simple differential equations, we talk about what a solution is. So they could take a solution, take the derivative and check that it's a solution, but we're not teaching them how to find solutions because, you know, especially in that last unit, things are getting complex enough. We're just having them do things numerically. A lot of our students will maybe take a differential equations class in addition to that, but we didn't want it to be too similar. Yeah, but I will say I teach differential equations a lot, and I have used some of the material in that course as well.
Right. But I would say that that's definitely the case. We have probably half the students in the class might have had differential equations, and the other half definitely have not. So you've got all levels of sophistication, but it turns out that the modeling itself and thinking very simply turns out to be hard. Sometimes you think, well, I've got this really simple problem of logistic growth. Like, what's the problem? Why are we thinking about it? But when they start doing step by step by step, even the students with more mathematics have trouble. And so we move through that pretty quickly. But how do I explain this? By doing it holistically, I think they're learning much more thoroughly than if you put them, we've got to learn all these tools first, and then we're going to apply them.
Awesome. Thank you so much. I think this is really amazing. Thank you.
Yeah,
Thank you.
We got one more question in the chat. It said, like, how many are the written? Okay, who is the general audience? Is it about AF Math, AF Bio students? What year are most of the students in the class?
I think it depends on which course, but in the kind of original course and the one that I'm still teaching, it's a maybe 52% math majors, something like that. The rest bio or environmental studies or chemistry majors. And we're beginning to see a lot more double majors taking the class. So that's the audience. And the audience, there's a rare first-year student, sophomores and juniors, and a rare senior. But Becky, you've used it in a number of different ways.
I have. I just taught a special topics modeling course last semester using this book, and it was, sophomores through seniors, but I got people majoring in biology, chemistry, engineering, computer science, math. It helps too if I was able to get it to count as a quantitative elective for the biology major. Like if you can get it to do things like that, like talk to your colleagues, that can really help attract students to the course if it counts for something in their major. But I've also adapted it to an upper level math course. I just might spend more time on like the numerical methods or the local sensitivity analysis, right? Just get more into the mathematics of those things. So I've actually used these case studies just in different ways